I think we're at a pretty stable number now. So let me begin. So hello, and thank you for joining us today for our webinar, 6 Steps to Success in Discovering Monoclonal Antibodies Against Multipass Membrane Proteins. My name is Joe Rucker, and I'm the Vice President of Research and Development here at Integral Molecular, and I will be the moderator for this webinar. Let me just give a few basics before we begin. The 2 presentations today will total about 40 minutes with a live Q&A at the end. Please ask any questions using the Q&A section on your screen. It's in the lower portion of your screen. There's a thing that says Q&A. That's where you can type in your questions. And let me begin. So I'll first introduce the speakers, and then we can get the webinar properly started. So our first speaker is Sharon Willis. Sharon is the VP of Technical Sales and Customer Support, and also a co-founder of Integral Molecular. Dr. Willis has been involved in all aspects of product development, from research and application development in the lab through manufacturing, quality control, sales, and customer support. Her expertise has been instrumental in launching the company's products and successfully placing Integral's technologies with hundreds of customers worldwide, including all of the top pharmaceutical and biotechnology companies. Dr. Willis will discuss lipoparticles as a source of high-quality membrane proteins for antibody discovery. Our second speaker is Ross Chambers, who's our Vice President of Antibody Discovery at Integral Molecular. Dr. Chambers directs all antibody discovery projects at Integral Molecular. He brings a decade of experience in the industry, leading the discovery of more than 2,000 antibodies. Dr. Chambers pioneered the use of DNA immunization for the production of antibodies in 2003, commercialized the genomic antibody technology, the GATT technology, for the rapid isolation of antibodies, and he continues to develop new antibody discovery technologies. Dr. Chambers was previously the Director of Research and Development at SAIX, where he directed the discovery of thousands of commercial antibodies using DNA immunization, B-cell cloning, and other isolation techniques. And before that, he developed DNA immunization for large-scale antibody isolation projects while at UT Southwestern. Dr. Chambers will discuss how Integral Molecular's antibody discovery platform can allow for discovery of antibodies against the most challenging membrane protein targets. Before we actually begin, let me actually introduce Integral Molecular as a company. So for those of you unfamiliar with Integral Molecular, we are the leaders in antibody discovery. Located in Philadelphia, we have close to 20 years of experience in the discovery and characterization of antibodies against the most challenging membrane protein targets. We have worked on thousands of different membrane protein projects and have experience with the most challenging, with most classes of membrane proteins, including GPCRs, transporters, and ion channels. And with that, I will hand over the talk to Sharon.
Thank you, Joe. Next slide, please. Thanks. Thank you, Joe, and thank you everyone for attending the webinar today. Multipass membrane proteins are really great targets for antibody therapeutics, particularly in oncology, immune modulation, and chronic inflammatory diseases. As shown in this figure, taken from the 2018 review by Carter and Lazar, looking at the top center circle in red, focusing on target biochemistry for multipass membrane proteins is a key for drugging this high-hanging fruit. Next slide, please. So let's talk a little bit more about drugging the undruggable. There are 2,400 multipass membrane proteins in the human genome, and so far only 3 of those proteins have approved antibody therapeutics. So why is this? A lot of it has to do with their complex structures, small exposed extracellular regions, and the overall structural dependence on the lipid bilayer. These proteins also tend to have low immunogenicity due to high sequence conservation in mammals and are poorly expressed or very toxic when overexpressed, thus presenting a lot of challenges for a successful antibody discovery campaign. Next slide, please. So today, Ross and I are going to talk about the 6 steps to success in discovering monoclonal antibodies to multipass, membrane proteins, and in the process, address 13 key considerations for these important targets. The 6 steps addressed start with antigen, and specifically antigen expression, the ECD size, as I mentioned before, sequence conservation, and always, I'm going to reiterate this many, many times, maintaining native conformation. Next is diversity with respect to the choice of animal for immunization, so that the project delivers a diverse panel of antibodies. Next is conformation, specifically always maintaining native epitopes, because that's really critical for a successful campaign. Next is function. The discovery of functional antibodies really requires large panels of antibodies with diverse epitopes. Next is specificity, so making sure that the leads that you generate are monospecific and only hit the target of interest. And then finally, developability, considerations, which include affinity and humanization. Next slide, please. So this is an overview of the technologies that have been developed and are offered by Integral Molecular for the discovery and characterization of antibodies targeting membrane proteins. With our antibody discovery platform shown across the top in that orange line, we can go from target discovery and antigen presentation all the way through antibody discovery, characterization, and engineering. Much of the success of our platform is enabled by lipoparticles, which present antigen in its native conformation. And again, I'm going to say this is a critical factor in any successful antibody discovery campaign. We also have two high-throughput cell-based platforms that are used in the discovery and characterization process. The first is shock immunogenesis, which involves making comprehensive alanine scan libraries that can be used for antigen engineering if that's needed, and more commonly for amino acid resolution epitope mapping on the characterization side of things. And finally, our membrane proteome array for specificity profiling of lead antibodies, as well as target discovery. And Ross will talk a little more in detail about our membrane proteome array. So these technologies together enable our high success rate of greater than 95% for discovering antibodies against membrane proteins. Next slide, please. Let's get started talking about the 6 steps to a successful campaign. Step 1 is working with optimized native antigen and addresses the problems I previously stated here on the left. Next slide, please. There are many choices for antigen to consider when starting a campaign. There are peptides, which can work well for making reagent antibodies, such as antibody for Western blotting. But overall, the antibodies generated from peptide immunization tend to be low affinity and do not see conformational epitopes. Next is purified protein. The main challenge here for membrane proteins is maintaining the native conformation of the protein, which again is critical for a successful project and hard to do with a purified membrane protein. Cells can and have been successful, but have their own challenges. And as Ross will discuss in more detail, we find using a combination of DNA and lipoparticles for immunization gives a robust and reliable immune response. Importantly, both antigen sources maintain the native conformation of the protein of interest. The choice of antigen impacts many important aspects of the project, including the immune response, antibody isolation, and lead selection. Next slide, please. So we usually start every project and recommend that you do as well with an assessment of the target. And the complexity of membrane protein targets requires some detailed homework up front to ensure that you get the antibodies you're looking for on the back end. These are large, long projects, and you want to make sure you do the work up front to help get what you need. So 3 areas to focus on are 1, specification of the antibodies you're looking for, the mechanism of action, are you targeting a specific epitope, and also thinking about the final therapeutic format. Next is antigen design, keeping a number of factors in mind. Certainly the overall structure and topology of the target, expression levels, higher is better generally, paralogs and splice variants. This is important when thinking about specificity, and then also epitope accessibility, just to name a few of these. And finally, the antigen format and production of the antigen. Are you going to use virus-like particles such as lipoparticles, soluble protein or peptides, cells, or something else? It's also important to find out what commercial reagents, if any, are available to QC the antigen. How will you detect the antigen that you make and know that it's good for your immunizations? So really, a customized, well-thought approach leads to success. Next slide, please. All right, we're going to talk a little more about developing an optimized antigen. The suggestions and considerations on this slide will help overcome many of the expression problems associated with the membrane proteins that we all work with. First, it's important to work with an optimized expression plasmid. Next, make sure your construct is codon optimized. We found this to be very helpful in expression. Engineering may be necessary to remove toxicity, improve expression, and possibly to remove or alter an immunodominant element in the protein to broaden the epitope coverage. But the challenge here, the real challenge, which our molecular biologists take on almost every day, is to engineer while maintaining native epitopes. That's still critical, even during engineering. And finally, consider using translation and trafficking enhancements in your constructs. And this is all because high expression is a key for a robust immune response and efficient antibody isolation. Next slide. So I'm going to end step one talking in a little more detail about lipoparticles. So lipoparticles are virus-like particles that we can engineer to contain high concentrations of the target membrane protein in its native conformation. The particles have a viral core protein surrounded by a lipid bilayer containing the membrane protein of interest. They're made by transient transfection. Typically, we use either hex cells or an avian cell line. The membrane protein of interest is overexpressed on the cell surface. The viral core protein assembles and buds through the cell surface, pinching off a piece of the lipid bilayer containing the overexpressed target protein. Then the lipoparticles are purified from the culture supernatant. Advantages of lipoparticles include native conformation and presentation of the membrane protein target, which again is critical for a successful campaign. The lipoparticles themselves are highly immunogenic. They're virus particles, and our immune systems or animal immune systems are really targeted to viruses. And the target protein is concentrated on the surface of the lipoparticles. So again, high concentration, native conformation protein leads to good immune responses. And next slide, please. Lipoparticles really are a leading solution for membrane protein antibody discovery. We have been making lipoparticles for over 20 years and have numerous protocols and SOPs for production, purification, and QC, leading to a very high quality, reliable reagent. 88 applications using lipoparticles, and we've worked with over 300 customers in our 20 years. All this together leads to our very high success rate, greater than 95% project success rate in incorporating really hard proteins onto the surface of lipoparticles, including GPCRs, transporters, ion channels, and other membrane proteins. And fun fact, we have made over 2,000 lots of lipoparticles. So that is it for my part of the talk. I'm now going to hand over to Ross to talk about the next steps for a successful campaign.
Great. Thank you, Sharon. Yeah, so if you remember one thing today, it's the importance of quality antigen in your campaign. Okay, so now you have a quality antigen of some kind in hand, and you then want to move on and discover antibodies with it. So the key problems here is ensuring that you could generate diverse range of antibodies to the target. And we'll talk about our solutions here. we have worked on antibody discovery on membrane proteins for many years here. It's all we do. We focus on this class of proteins. And we have made all the mistakes, discovered all the problems over the years, which we're sharing with you today. What we have discovered, we've developed a number of technologies and assembled it together in this platform that we call the MPS Antibody Discovery Platform that enables us to have this high success rate against a diverse range of membrane proteins. And so just briefly, our system that I'll walk you through and some of the solutions that we have discovered are things that such as DNA immunization with Lipoparticles into a diverse range of species, isolation of those antibodies from those animals using Lipoparticles in phage display, and then able to humanize and characterize those antibodies to find those lead candidates such as antagonists, agonists, etc., that you need for your therapeutic. Okay, so first a word about how we discover antibodies. I mean, we rely on animals, and I know there's many choices for discovering antibodies out there, but we have relied on animals not just because of the usual advantages of higher affinity antibodies and developability of those antibodies, but what we have found is that really it's been the most successful for multipass membrane proteins, and we get very diverse ranges, panels of antibodies out of animals, and they're able to cope with the much lower levels of expression relative to a typical soluble protein. Now, one of the great things about using animals is there's a technology that Sharon mentioned is DNA immunization. And this is really an ideal technology for multipass membrane proteins. And for those not familiar with this, DNA immunization is the process where you deliver a plasmid coding your target antigen into an animal, transfecting the cells inside the animal where they display the antigen for the immune system to respond to. And why this is ideal, of course, for membrane proteins is these are very fragile, difficult proteins, and they're displayed in their native context. Now, one of the tricks that we've discovered over the years with DNA immunization is often you don't always see a titer after immunizing with DNA. I mean, sometimes you do, but in many cases, you do not see any titer. However, underlying that is there often is almost always is an underlying memory B cell response. So the animals have responded to your antigen, but just laid down memory B cells and not produced antibody. So what we have found and others is that giving a small amount of protein to those animals triggers those memory B cells to convert into antibody producing cells where you can now see a strong, robust titer. And so our experience has been that combination of DNA and protein has been very effective at producing strong immune responses against native extracellular epitopes. Now, while we're big fans of DNA immunization because of its, as I said, the native presentation of the antigen, and it's a pure antigen, only one thing is displayed, I would say that, you know, it's not always trivial to use DNA immunization. There's many practicalities to deal with. So to really optimize this, to make it work well, you've really got to think about the design of the plasmid, the design of the gene, the design of all the elements on that plasmid to ensure that it's highly immunogenic when you deliver into cells. It often requires the addition of adjuvants to work well, often genetic adjuvants where you're delivering genes on additional plasmids to boost the immune system. You have to think carefully about how you deliver the DNA to ensure the animals respond, as well as boosting, as I mentioned. And so, as I mentioned before, boosting with protein is highly effective at making this work well. And then I was an early pioneer in the DNA immunization field, and we showed many years ago that adding DNA adjuvants has a profound effect on the titers of DNA immunized animals. Now, as you may see in those previous pictures, chickens figure prominently. And the reason is a problem that we have encountered and many others have encountered is how do you access conserved epitopes? Now, you may be aware that many human proteins are highly conserved across mammals. So in fact, half of all drug targets are more than 90% identical across mammals. And so if you immunize a human target into mice, you'll often get a very weak response. Now you can go to other animals such as rabbits or llamas, but really that has a marginal benefit. All mammals are relatively closely related. And so what that has led us to is to focus on chickens, because chickens have a much greater evolutionary distance. And in fact, if you compare the proteomes across humans and chickens, in fact, most drug targets are not highly conserved in chickens, and you get robust responses. And so this enables us, using chickens, to access conserved epitopes. And an additional benefit is your ability to generate pan-reactive epitopes, so antibodies that recognize not just the human version, but also the mouse and the monkey, to enable preclinical studies and not have a surrogate. Okay, so just some data to show how well this works. So first, how well the chickens work with generating antibodies to highly conserved targets. So here are 4 targets that we have worked on in recent years: Claudin-6, KV1.3, CB1, and GLUT4. All of these are complex membrane proteins, and all of them have greater than 95% identity to mouse, meaning that immunizing to mice is going to be a real challenge because of immune tolerance. However, when you compare that identity to chickens, you see this much lower identity, and it's down at levels where now generally immune tolerance is not a problem. And here's some examples showing that when we immunize these highly conserved targets into chickens, we get robust immune responses to them. Now, I mentioned an additional benefit is the ability to generate pan-reactive antibodies, and this is a feature that we often identify in our campaigns. So here again is 4 targets we've worked on recently, and we immunized with the human target, and we saw strong, immune responses in the animals. But when we test that same serum on the rodent paralog, we see strong reactivity to those to the rodent version. So surrogate antibodies are often not required from the using the chicken system. All right, so you have immunized animals, you've generated high titer responses. The next step is isolating the antibodies from those animals. And here it is key to make sure that you focus in on the native epitopes. So depending on how you immunize, you may generate some native epitopes, but you may also generate a lot of irrelevant things that you may have to sort out. But you want to focus in on those native extracellular epitopes. And for that, we use phage display with native antigen. So a key thing with phage display is having high concentrations of antigen for the panning. So with phage display, you generate these very large libraries. So we will create large 1E9 libraries from those immunized animals using a variety of tissues. And you want to efficiently enrich those antibodies out of there. And for that, high antigen concentration is key. So for that, Lipoparticles have been very valuable for us. It represents a very highly concentrated form of the antigen and enables that efficient enrichment of the antibodies. So typically for our campaigns, we will make these large immune phage libraries. We will deselect out non-specific MAbs and undesired cross-reactive MAbs if required, and then pan on those on Lipoparticles to efficiently enrich diverse ranges of antibodies. And then after panning, of course, you need to go screening. And for screening, you need to again think about native epitopes. And for that, we use Lipoparticles again in high throughput ELISAs and in flow cytometry to detect antibodies that focus on the native extracellular epitopes. All right, so now we get to screening. So for success in a campaign, and especially for finding functional antibodies, the really key things for finding functional antibodies is just getting a large number of antibodies and making sure that they recognize diverse epitopes. And so that means is a lot of screening, and screening is very labor intensive. So what I want to talk about now is what we have done to address this, which is developing automated deep screening. So what we have done, so after panning and those phage libraries, and we've got many, many colonies, is we have developed the capability with a lot of robotics and automation, is to perform thousands of immunoassays to screen the SCFE peripreps from those phage clones. Then once we've identified those hits, we'll use the robotics again to pick all those clones and format them. And then for functional antibodies, we often need purified IgG. So we've developed a system that enables us to subclone on a high throughput basis hundreds of genes and into an IgG format, and to express and purify hundreds of IgGs. Okay, so you've generated hundreds of antibodies, hopefully, to your multipass membrane protein, and now it's working through the funnel to get to your lead candidates. And there are many things you need to test to find your lead candidate here that are listed here. But the key one I want to mention is functional assays. So if you're looking for a functional antibody, they're often very rare, and that is the key assay that chops down the funnel to a few lead candidates. So I want to talk about functional assays here. So the programs that we work on, a lot of functional programs here for these multipass membrane proteins, and you can divide these into two different mechanisms. And functional MAbs are rare because of these rigorous requirements to generate functional antibodies. So in the case on the left, where you want to, there are targets where you need to lock a certain conformational state in order to get an agonist or an antagonist. They're difficult to find antibodies to because you need to hit a very specific epitope. And you also have to be able to display that conformational state to access. And often those sites have restricted access. Often they're in pockets and are difficult for antibodies to reach. Now, the other case for a functional antibody is when you block a ligand. And there, again, it's relatively difficult to find these antibodies because, again, you have to hit a very specific epitope, the ligand binding site, and you often need a high affinity antibody to compete with the endogenous ligand. Now, we have done many programs here successfully discovering functional antibodies, as shown in that left panel, and we've developed all kinds of antagonists, agonists, inhibitors to various proteins. And we routinely are successful at doing this, and for many of the reasons that I've described previously of getting large diverse panels of antibodies. But I want to highlight one other feature that we think chickens help in the success rate, and that is that chickens also produce antibodies with longer CDRH3s. And why this is important is that antibodies with longer CDRH3s form a protruding topology, and that enables them to access these pockets where often functional sites are. And this has been well described with llama antibodies, where they also have these longer CDR3s, and also some human antibodies have been demonstrated for various HIV antibodies. But these longer CDRH3s enable you to access these functional sites. It's been shown many times in crystallography with these longer CDR3s, as shown on the right-hand side. All right, so you have your lead candidates, and a key feature of antibodies, of course, is the ability to bind to a single target. But it shouldn't be assumed that all antibodies are monospecific. In fact, it's a common feature for them not to be monospecific. So you really need to be able to test them for monospecificity. And so the solution that we have come to here is the ability to do comprehensive specificity screening. So for those that are not aware, so really antibodies cannot be assumed to be monospecific. There's not an intrinsic feature of antibodies. They tend to be monospecific, but it's not you can't assume it to be the case. In fact, we have discovered and others that up to a quarter of antibodies are polyspecific, binding to more than one target. And not just things that are related by sequence, but also just irrelevant, unrelated things that just the antibody happens to be able to bind. And of course, off-target binding is a key issue when it comes to drug failure. In fact, off-target binding is one of the major factors involved in failure of drugs in clinical trials. So the solution that we've developed here at Integral is the membrane proteome array. What this is essentially the entire human membrane proteome displayed on an array, 6,000 targets, that we can screen efficiently using high throughput flow cytometry. Now, importantly, the antigens in the 6,000 array collection are expressed in cells, live, unfixed cells, so that the native epitopes are displayed. And in this way, we can rapidly determine whether an antibody is monospecific. As we've shown on the right here, this is one of our programs for Claudin-6. We've shown that it's monospecific in not binding to very closely related molecules like Claudin-9, which only differs by a few amino acids, or any other Claudin or any other membrane protein. All right, so we have a final candidate. Now, for all animal derived antibodies, of course, you have to humanize them so that they don't form immune responses in humans. And often that can be a problem because humanization step itself can result in loss of affinity or cause developability problems. So for that, we have developed a technology we call the humanized chicken antibody technology that I'll describe for you. So just briefly review what you need to do with humanization is often you have an animal antibody that you then want to humanize. So you'll CDR graft the loops, the CDRs onto a human framework. Often people will introduce back mutations to optimize that affinity. And often people will have to do affinity maturation by site-directed immunogenesis across the CDRs to improve the affinity. Or if the light chain is misbehaved, a light chain shuffle may be required. Now, all of this engineering takes considerable time. It's not trivial to undergo these steps. So what we have developed here recently, just this year, we've launched this, is that instead of isolating antibody genes from the immunized animal, we will isolate the individual CDRs. And we'll clone those as a pool directly from the animal into a humanized framework to make an scFv library. So this is a humanized framework with the animal CDRs spliced in. And then that library is then used conventionally for panning and screening to isolate antibodies. And what this means is that straight out of panning, you will be isolating fully humanized antibodies. These antibodies are humanized prior to MAb isolation. Now, an inevitable consequence of doing this is that the CDRs get shuffled. And we discovered somewhat unexpectedly that this shuffling of CDRs actually enables affinity maturation of the antibodies, and actually picomolar binders are routinely isolated from these kinds of libraries. Now, just to demonstrate how well the humanization works, one way to measure humanization is using the tool, the IMGT domain gap align tool. And here we've analyzed a large panel of clinical antibodies, some human, some mouse humanized ones, and to show the level of humanizations. You see that distribution of humanization in current clinical antibodies. The antibodies that we have isolated using the HCAT technology is shown in that red box, and all of them land within the 91 to 95% in the domain gap align algorithm. So we achieve a very high degree of humanization with this platform. Now, I mentioned the somewhat unexpected side benefit of affinity maturation by shuffling the CDRs, and this was demonstrated here. So here we did a campaign against Claudin-6, and we pulled out a large panel of antibodies using HCAT. And here in this graph, we're measuring the relative affinity of all these clones, and all of these clones shown here share the same CDR3. So they have identical CDR3s. And you can see a range of affinities. But what was really informative is when we went back into the chicken to find the parental antibody where all of these antibodies were derived from before the HCAT process. And when we measured the affinity of that, it was significantly lower. So we have generated significantly higher affinities with all of this recombination CDR shuffling. Okay, so that's a summary of the 6 steps. What I hope you've learned today is that you can be successful with membrane proteins, but it isn't a trivial thing. There's many things problems to address, but if you focus on these problems and come up with solutions to address them, you can be successful. We have applied this platform, our solutions, to a wide range of antigens, and we have shown that this platform gives a high degree of success rate against a very broad range of proteins, including some very challenging multipass membrane proteins. Okay, so just as a parting point, we talked to a lot of customers, and many times customers who've been working on membrane proteins have tried to solve it themselves with conventional approaches. And invariably, they've spent many years trying to develop antibodies and with low success rate because you're using conventional approaches. So what I want to tell you here is that you really face up to these issues and develop solutions to them. We have shown that you can get it right the first time, deliver antibodies within less than 1 year with a very high degree of success rate. All right, that's the end of our story. I'll hand it over to Sharon.
Yeah, thank you, Ross. So There's a number of ways of that we work with our customers. We have our reagents and services as a fee for service. That includes our epitope mapping, specificity profiling using the membrane proteome array, and purchasing lipoparticles. I also want to mention on the lipoparticle front, we will start chicken immunizations with a lipoparticle purchase if you're doing antibody discovery. A lot of people use this as a backup plan so that if your discovery plan doesn't go well or doesn't yield exactly what you're looking for, or if you're looking for a backup, we will have the chickens immunized and going and store those tissues for you in case you want to convert to an antibody discovery project. We do our antibody discovery projects on a partnership basis. We can also do novel target identification on a partnership basis. And then we didn't go through this, but we have our own therapeutic pipeline of antibodies that are available for licensing. Now I think we will go back to Joe for our Q&A. And again, if you have any questions, please put them in the Q&A at the bottom of the screen.
Great. Thank you, Sharon and Ross, for the webinar and all that the great information. So we're going to start the question and answer portion of the webinar now. So first of all, remember, if you have any, we already have a lot of questions, but if you have any more, please remember to put them in the into the Q&A box, and we will get to as many as we can in the time we have. And if we don't get to your question, you will receive an email to answer your question, as long as you didn't ask it anonymously. And of course, if you have additional questions, feel free to contact Sharon or any of us at info@integralmolecular.com. So let me begin with a question for Sharon. So how do you know that the conformation of the protein on the lipoparticles is native?
Yeah, that's a very good question. That comes down to that setup slide that I had where you do the assessment of the project. And it's important to have some kind of reagent, most preferably some kind of conformation dependent antibody that can be used on the lipoparticles once they are purified. So that's what we always aim for. Of course, there are, and then we look at that antibody binding by ELISA. That's what we always aim for. Those reagents are not always available, so sometimes that's not possible. But sometimes if there's like a fluorescent ligand or some kind of other reagent that we can use for detection, we will use that for QC of the lipoparticles.
Great, thank you. So the next question is a question for Ross. So the question is, there is a notion that antibodies are too big to access the loops of a GPCR. Is that true?
That the antibodies are too big to access the loops. That's not been our experience. I mean, we have, membrane proteins in general are somewhat challenging because their loops are small. They're very small. There's really not much to access, but we have done a broad range of different targets with very small loops. And our experience has been that antibodies can do it. They can achieve this and bind to these very small loops very effectively.
This next question is actually for Sharon, which is there a method, or do you have a method to control membrane protein concentration on the lipoparticle surface?
So not specifically, but what we do through our construct design is use all of those elements to really push as much protein as possible to the cell surface, which then enables the capture of a high concentration of the protein onto the surface of the lipoparticles. So our target, in working with this for a large amount of time, we've come to realize that if we can get 50 picomoles of specific receptor protein per milligram of total protein onto the surface of the lipoparticles or higher, that really leads to success in many of the different assays, antibody discovery, characterization that we are doing and that our customers want to do. So really our goal is to hit that 50 picomole per milligram mark, or hopefully even higher than that.
Great. This next question is for Ross. Have you tried the DNA plus lipoparticle approach in other species? And, is it superior? And if you did this in other species as well, since you just were talking about chickens here?
Yeah, it's a good question. No, I've actually, I've been working with immunization, DNA immunization for more than 2 decades. And I've used a lot of different animals. It works in mice and rabbits and goats and you name it. With immune system, prime boosting works very effectively. It's really that memory B cell response and converting it into antigen secreting cells with the small amount of protein. It works very well.
And sort of a follow-on question for that is, for DNA immunization, have you found that adjuvant addition works better or worse for different classes of proteins?
I think I'd say it generally works across many different targets. I mean, there may be cases where it works selectively, but it'll be hard to tell. We haven't done any specific cases, but generally we'll add these adjuvants and we've seen them broadly work across many different targets because of the mechanism of how they work. So many of the mechanisms of how these adjuvants work is fundamentally on the immune system, so they should work across multiple proteins.
Great. So this next question is, I think, a question for Ross, although Sharon may be able to comment as well, which is the question is sort of a multi-part one, which is do lipoparticles only carry the target of interest? Are there other proteins present? And really, if there are other proteins present, how does this affect, for example, immunization or affect your strategy towards panning?
All right, well, I'll answer the first part and then Ross can answer the second part. So for the first part, yes, there are other proteins on the surface of the lipoparticle. So the lipoparticles bud from the cell surface, and while the overexpressed membrane protein is there, again, at that 50 to 200 picomole per milligram concentration, there are other membrane proteins there. One of the ways that we control for that is that we also sell and include with orders what we call null lipoparticles. So those are particles made from the same cell line, but without the overexpressed membrane protein of interest. And then I'll let Ross comment on the actual benefits of those other proteins in immunization and then how to deal with that in panning and screening.
Yes. Yeah, actually, this again speaks to the strength of DNA priming and lipoparticle boosting. So with DNA priming, you're priming with a pure antigen. So even though there are other proteins on the lipoparticles, when you deliver the lipoparticles, they're only priming them, so you get a very weak response to those other proteins. And the focus of the immune system is on the target. I would say also that it's a very useful positive control. So we do get a small number of antibodies to those unrelated proteins on the surface. Is not generally a problem, but actually is a useful internal control because we always like to see them come up on our screens. It means that all the technical steps are working, that it's validation that your library and panning and everything is working when you see those come up. So it's a valuable internal control as long as they don't become a predominant problem. And they generally are not a problem because of the DNA prime lipoparticle boost strategy that minimizes the generation of responses to them.
Great. So the next one is pretty much is for Ross, and really, can you comment on how you talked about antibodies with these long CDR H3s, and how does the stability of those compare to antibodies with moderate lengths of CDR H3s?
Yeah, it's a good question. There have been quite a few antibodies discovered with longer CDRs, not just from chickens, but llamas and humans as well. And the general experience has been they are quite stable antibodies. We've never detected any major issues with longer ones. We've seen no correlation with stability or aggregation or any other issues with them because the antibodies have evolved in an animal and are selected for stability because they come through an animal. I think it'll be more of a concern with a naive library because they haven't gone through that selection for stability. But I think the ones we pull out from antibodies are very well and stable antibodies.
And so it's just sort of a follow-up question then, which is, someone asked, do the CDR shuffled clones coming out of the HCAT platform have a poorer developability profile than parental antibodies coming from a natural chicken?
Yeah, we haven't seen that. It's a potential concern. although we do design the way we assemble the libraries in a special way to minimize any issues. But what our experience has been that these look fine in stability. We do thermostability, SEC for aggregation. We haven't seen any problems. In fact, some of our finest antibodies have come out of this technology.
The next thing is really almost a subject for another webinar, but I'll direct this towards Sharon, which is about the membrane proteome array. Although Ross can comment, in the off-target array, do you get problems of high background binding since the cells are not fixed?
No, we actually we have not seen that at all. I don't think.
Yeah.
So not the importance of not fixing the cells is, again, maintaining those proteins in their native conformation. If there are any background issues, we can work with the membrane proteome array in alternate cell types. So every project starts with an assay setup so that we can see how well your antibody is performing on our cells and our system. If we notice any high background binding at that point, we can take care of it at that assay setup stage so that we're confident when we go onto the array with the unfixed cells that the results are clean and clear.
Great.
And we have seen loss of reactivity with fixed cells. I mean, that's one of the things we learned early on, is that if you fix cells, you lose some of that reactivity. Some epitopes are destroyed inevitably.
Yeah.
And actually, somebody also asked a question about the membrane proteome array, whether the membrane proteome array has been used for IND submissions.
Oh, yes, it has. Yeah, we supply IND ready material with every project.
Great. Okay. The next question is actually for Sharon, which is really sort of a more general question. Have you had any issues expressing any dimeric or tetrameric membrane proteins when making the lipoparticles?
No, we have not. We have actually made some quite large complexes, multimeric proteins, and incorporated them well into the lipoparticles. So that really is not a concern. And again, every lipoparticle project for a customer project is staged. So we do an initial assessment of the expression of the target, whether it's monomeric or multimeric, and make sure that we can detect all of those components in that kind of setup phase of the project before we go on to the large scale production and QC and purification. So that way we can catch any problems early on, and if necessary, go back to the molecular biology to re-optimize constructs to get either better expression or better incorporation, and make sure that we are expressing and detecting exactly what is needed for the project.
Great. This next question is actually also a question for Sharon. Is how do you determine off and on rates using lipoparticles, for example, with a GPCR?
Okay, yeah, very good question. So lipoparticles, they're actually really little handy little tools. So they can be attached to a biosensor surface. We've worked with a number of different BioCore models. We currently have an octet on site. So lipoparticles can be biotinylated, so they can be captured to a streptavidin chip or surface. There's other ways to capture them as well. And then you can flow the antibody across the captured particles to determine on and off rates. And we've done this with both full IgG and with Fab.
Great. So I think what I want to do is end with two final questions. The first question here will be for Sharon, which is how stable are the lipoparticles?
Again, as I said, these are great little tools. They're very stable at both 4 degrees and -80 degrees for a few years. We have data going out, a number of years. The only issue with stability is with detergents. So, you have to be very careful, since they do have that lipid bilayer. They are very susceptible to detergents. So low levels of tween, which are often used in panning, and then also like in biosensor experiments, are okay. But other detergents are very harmful. We've actually published a paper on this, and if you send an email, you can email any questions to info@integralmolecular.com. We can send you that paper that we published looking at the detergent stability of lipoparticles.
Yeah, I would say also as an in-house customer, we find them very useful in our campaigns. We make a large batch up front and it gets us through the whole campaign. I mean, generating cells and things is logistically complicated, a lot of steps to organize, and having something in the fridge ready to go makes all the workflow much smoother and more efficient.
Yeah, Ross is one of our best customers.
And then a final question for Ross, which is really can, obviously you have a very sophisticated technique here, but can peptides be successfully used to generate antibodies against membrane proteins?
Yeah, we get this question a lot because, peptides, as you know, are cheap and easy to get hold of. But really, and I've tried them many times over the years, and really they're not really a source of success here for multipass membrane proteins for native epitopes. And the reason is simply that peptides don't form structure. And you'll get antibodies that recognize the protein on Western blots, but I can't think of a similar example over 2 decades where they've worked well. There's always a person that had one success story, but really it has been a very poor channel for success is peptides. I mean, they're great for other things, but really for this area, they haven't been an avenue for success.
Great. Thanks. So, people can continue to put in questions, which we can get to by email, but that's really the end of the questions we have now. So first of all, I want to thanks again to our speakers, Sharon and Ross, for their informative presentations and for answering all the questions from the audience. Again, if we didn't get to your question here, you will receive an email, or you can email us at info@integralmolecular.com and make sure that your question gets answered. And then finally, I want to thank you all for joining us today. And that ends our webinar. So enjoy the rest of your day. Thank you.
Thanks, everybody. Have a good day.
Bye.
Bye-bye.